Dr. D, 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 Dr. D. Explain stuff.
Hey everyone, Dr. D here, and in this video we are going to be covering Chapter 2 from our Genetics Essentials textbook, Concepts and Connections, 4th edition. This chapter deals with chromosomes and cellular reproduction, so let's go ahead and get started. We're going to be touching on the difference between prokaryotes and eukaryotes. We're going to be talking about cell reproduction, sexual reproduction, so that's going to entail parts of mitosis and meiosis respectively. Now, I want you guys to remember the differences between prokaryotic and eukaryotic cells. I have a nice video on the YouTube channel from my Biology 1406 class, Chapter 4. If you need a refresher for the difference between prokaryotic and eukaryotic cells, and you need a refresher for the parts of the cell, what is a nucleus, what is mitochondria, etc., etc. Chapter 4 for Biology 1406 is where you'd want to go. So let's break it down. A prokaryotic cell is a simple cell. It's usually much smaller than a eukaryotic cell. Remember that prokaryotic cells lack a nucleus, and they also lack any other membrane-bound organelle. But they do have DNA. You see this stringy blue stuff? This is DNA. You need to know that prokaryotes have usually one chromosome, and it's a circularized chromosome. That means there's no beginning to the chromosome, there's no end to the chromosome. It's double-strand DNA, but it's a circle. It's a closed circle. Think of a rubber band, right? A closed circle of DNA. And that DNA does not have a nuclear envelope around it. So that DNA is just floating around in the cytoplasm, and it's considered the nucleoid. Now, you don't have membrane-bound organelles inside of a prokaryotic cell either. So you don't have mitochondria, you don't have chloroplasts, you don't have a nucleus, you don't have endoplasmic reticulum, you don't have Golgi apparatus, etc. What you do have are some organelles that are non-membrane bound, like these pink or peach-colored dots. You see these little pink dots? These are ribosomes. You do have ribosomes because ribosomes are non-membrane bound organelles. And remember, what type of cells are prokaryotes? Bacteria, the domain bacteria, and the domain archaea. Those are the two domains of life that are comprised of prokaryotic cells. And remember this from Biology 1406. Bacteria and archaea, essentially the prokaryotes are single cell creatures. So the whole creature is one cell. Now, eukaryotes, on the other hand, these cells are much bigger, right? You've got the plant cells, you've got animal cells. These are eukaryotic cells. How do I know? Well, I see a nucleus. That's a giveaway right there. That's a telltale sign of a eukaryotic cell is the presence of a nucleus. What is a nucleus? That means that the chromosomes are inside of this double membrane called the nuclear envelope. So the nucleus itself is considered a membrane-bound organelle. It is the main membrane-bound organelle. And then inside of the cell, look inside of the cell, you've got a host of other membrane-bound organelles. You've got this convoluted membrane called the endoplasmic reticulum. You've got this membrane, this is the membrane of the mitochondria. You've got the membranes of the Golgi apparatus. You've got all kinds of membranes inside of the cell. In the plant cell, you also have the addition of the membrane of the central vacuole. You've got the membrane of the chloroplasts. So you can see here there are many, many membranes. By the way, before I forget, remember that all cells have a plasma membrane. The plasma membrane is the membrane around the whole cell that envelops the whole cell. So plant cells have a plasma membrane. Animal cells have a plasma membrane, and like I said, all cells have a plasma membrane, so prokaryotic cells have a plasma membrane as well. Again, if you need any kind of refresher on this, Chapter 4 from Biology 1406, I have a video on there on the YouTube channel. Check that out, and it's a comprehensive review on this material. Now, remember what I said. Prokaryotic cells, nucleus is absent, cells are relatively small. And the DNA is usually one molecule of one chromosome, and it's circularized. Okay. Oh, and I didn't mention histones. In prokaryotes, you don't have histone proteins, but in eukaryotes you do. Let me show you what that means. Here, let me show you what that means. This is what we're talking about when we're talking about a eukaryotic chromosome. You see, this is that X-shaped structure that you guys may know about, the eukaryotic chromosome. Well, like I said in chapter 1, this chromosome is not just DNA, is it? It's not pure DNA, is it? That's actually DNA. You see, DNA wrapped around these histone proteins in eukaryotes. These are proteins where the DNA wraps around. It wraps around a couple times, right? Now, what you need to know is these histone proteins, it's actually 8 different proteins. It's called the histone octamer. And this structure I'm boxing in right here is called a nucleosome. A nucleosome, that means 1 unit of DNA wrapping around the histone octamer, nucleosome. And together, the DNA plus the histone proteins make up your chromatin. Chromatin is the material that makes up your chromosomes, right? But like I just mentioned, in eukaryotic cells, your chromosomes exist as chromatin. That means DNA complexed with histones. In prokaryotes, except for some archaea, you don't have histones, but bacteria have something else. Usually it's called histone-like proteins that it wraps around. It's not exactly histones. Okay, the relative amount of DNA is small in prokaryotic cells. Remember I said it's a small circularized chromosome, whereas in eukaryotic cells you have a lot of DNA and it's linearized. What do I mean by linearized DNA? Human chromosomes, eukaryotic chromosomes, have a beginning and an end, right? There's 2 ends to the chromosome. It's like one long thing, right? One long double-stranded DNA. That's your chromosome. Whereas with prokaryotes, there is no beginning or end to the double-stranded DNA. It's like a rubber band, right? The double-strand DNA has no beginning, has no end, and there's only one rubber band, right? There's only one chromosome in a prokaryote. Whereas in you and me and in eukaryotes, the double-strand DNA has a beginning and an end, and there's many of them. There's many chromosomes. Humans have 46 chromosomes, right? You inherited one set from your mom of 23 chromosomes from your mom, and you inherited the other set from your dad. So again, prokaryotes, unicellular creatures with no membrane-bound organelles. Prokaryotic DNA does not exist in the highly ordered and packed arrangement, so it doesn't condense itself to the extent of eukaryotic chromosomes with their histones. And remember, prokaryotes make up the domains of life: bacteria, also known as eubacteria, and archaea. However, in eukaryotes, you have both unicellular and multicellular creatures that have membrane-bound organelles. So, did you know that there are unicellular eukaryotes? Think about algae. Think about the protozoa, right? You know, the protozoa, they include the trypanosomes, they include amoeba, they include all sorts of different single cell organisms, paramecium, for instance. So with eukaryotes, you have many examples. Usually these are called the protists. You have many examples of single cell creatures, but you also have many examples of multicellular creatures. Okay, there are single cell creatures and multicellular creatures as eukaryotes, whereas with the prokaryotes, you only have single cell creatures, right? An E. coli is it's one cell, right? Okay, so but like I said, you can have multicellular or unicellular cells, but the unifying feature is these membrane-bound organelles, specifically the main one, the nucleus. The genetic material is surrounded by envelope, the nucleus, the nuclear envelope, the nucleus, and the DNA is closely associated with histones to form tightly packaged chromosomes, right? Like this. Look how tightly packaged this DNA is, right? Look how tightly packaged the chromosome is. This is, if you were to unravel this chromosome, it would be several feet long, right? But it's all packaged into one little tiny bulky structure, condensed bulky structure. So let's do a concept check. List several characteristics that eubacteria and archaea have in common that distinguish them from the eukaryotes. Well, we just listed a host, right? Just think about it. Eukarya and bacteria and archaea are all different, right? The prokaryotes have, they're single cell only, they're smaller, no membrane-bound organelles. This includes the main membrane-bound organelle, the nucleus, right? They have one chromosome, it's circularized, right? And so that's plenty right there, plenty of examples right there. Now let's touch on viruses. I want to ask you, I think we touched on this in chapter 1, but are viruses alive? Think about that for a second. No, hopefully you answered no. A virus is not technically alive, and most biologists would agree that viruses are not technically living entities. They are what's known as small infectious agents, okay? They don't have enough of the properties of life to be considered living, okay? And in essence, essentially what a virus is some kind of genome, and that genome could be a DNA genome. Look at this drawing right here. This genome is DNA, but that some viruses don't have a DNA genome. They have an RNA genome. They have an RNA genome. So you have some kind of genome. Remember that genetic information can be expressed or can be stored as DNA or RNA. In viruses, you can have DNA genomes or RNA genomes. Either way, a virus is essentially a genome with a protein coat, a viral protein coat. And inside of, on top of this protein coat, you can have proteins such as spike proteins, for instance. You know, these are proteins that are on the surface of that coat. Now, when we're talking about prokaryotes, and we're going to delve into prokaryotic cell reproduction, you need to remember some concepts from Biology 1406. Do you remember the origin of replication concept? Remember, a prokaryotic chromosome is like a rubber band. It's circular. There's only one spot on that circularized chromosome where DNA starts to replicate, and that's called the origin of replication. So only at one spot will this circularized chromosome start replicating, and it replicates in both directions from that spot, right? That's called the origin of replication. But remember, eukaryotes have much longer linear chromosomes, and they would have multiple spots along that chromosome where DNA is going to originate its replication. So you could think of it this way. Eukaryotic chromosomes have multiple origins of replication when you're copying the chromosomes. Prokaryotes only have one origin of replication when you're copying the chromosome. And prokaryotes have a relatively high rate of replication of the whole creature, right? So, for example, an E. coli cell can double every about 25 minutes or so. And that's because they have relatively smaller chromosomes. They only have a small circularized chromosome to copy. You and I, our cells take hours to replicate, usually along the lines of 16 hours for our cells to replicate, at least for our cells to replicate. Why? Well, because we have much more DNA to copy, and we have a lot more that goes into mitosis. Okay, eukaryotic chromosomes. Remember, eukaryotic chromosomes, it's not just one circularized chromosome, is it? It's multiple linear chromosomes. In the case of humans, we have 46 chromosomes. We inherited 23 of those from mom. That's called a set from mom. And we inherited 23 of those from dad. That's called a set from dad. These are called homologous chromosomes. What does that mean? That means that they are not identical, but they have the same chromosome number. So if I inherited chromosome 1 from mom and chromosome 1 from dad, they're not identical in what they say. They're just identical in the chromosome number and in the types of genes that live on those chromosomes. So let me show you this. For example, if eye color is on chromosome 3, for instance, I'm just making something up off the top of my head. If eye color allele is on chromosome 3 from my dad, it would also be on chromosome 3 from my mom. But that does not mean that these are the exact same information. This could be brown eye information. This could be blue eye information. Does that make sense? So that's the way you should think about homologous chromosomes, is that you got one from mom, one from dad, or one from dad, one from mom. They're not identical. They are not identical. They have the same chromosome number. So if this was chromosome 3 from mom, this would be chromosome 3 from dad. And they have the same types of genes, which means eye color gene, hair color gene, insulin gene, the same types of genes, but they're not going to be identical, because you could get a brown eye color gene from dad and a blue eye color gene from mom. So think of homologous chromosomes as they share the same number, right? Chromosome 1, chromosome 2, chromosome 3. They share the same types of general information, but they are not identical. You got one from mom and one from dad. Okay, so if you think about homologous chromosomes that way, you're set. I have a really good video on homologous chromosomes versus sister chromatids, knowing the difference between the two. I'll show you that video, or I'll link to that video. And that's going to help you so much to understand mitosis and meiosis. If you're foggy at all on the difference between mitosis and meiosis from Biology 1406, this video is going to help you so much, because I've got a nice video where I just spend a lot of time explaining homologous chromosomes, explaining the difference between homologous chromosomes and sister chromatids. Okay, so we're going to talk about the cell cycle. Remember, this is interphase followed by mitosis. And then we're going to talk about the genetic consequences of the cell cycle. Remember, if we're talking about mitosis, the genetic consequences, 2 genetically identical cells. If we're talking about meiosis, the consequences, the gametes, right? The genetically non-identical sperm or the egg. Remember these terms from Biology 1406? They're coming back to haunt us. Diploid cells versus haploid cells. What does haploid mean? Let's start with haploid. Haploid cells carry one set of genetic information. Think of the sperm. Think of the egg. The sperm has one set of chromosomes, doesn't it? It only has one chromosome 1. It only has one chromosome 2. It only has one chromosome 3. It has 23 chromosomes. That's what haploid means. It only has one set of chromosomes. Okay, the egg and the sperm are haploid. Now, when the egg meets the sperm, right, when the egg is fertilized by the sperm, now you have 2 sets of chromosomes, don't you? You had the set that was in the egg, and then the set that's being delivered by the sperm. Now you have 2 homologous sets of chromosomes. Remember that term, homologous? In fact, you might want to write in your notes right here, this has to be homologous sets of chromosomes for it to be diploid. Fun fact, you can't just copy one set of chromosomes. You can't just copy a set of chromosomes and then call yourself diploid just because now you have 2 sets. They have to be homologous sets. One set has to be from your mom, one set has to be from your dad for it to be diploid, for it to be homologous sets and diploid. You can't just take a sperm cell, for instance, and then just double the chromosomes and copy all the chromosomes and then call it a diploid sperm, for instance. You could not do that because it would just be a sperm cell with 2 copies of the same set, if that makes any sense. Awesome. So let's do a concept check followed by a short break, and then we'll come back to tackle more of the chapter. Concept check number 2. Diploid cells have what? 2 chromosomes, 2 sets of chromosomes, 1 set of chromosomes, 2 pairs of homologous chromosomes. This is kind of a tricky one. What do you guys think? Think about it. 2 chromosomes, 2 sets of chromosomes, 1 set of chromosomes, or 2 pairs of homologous chromosomes. This is a tricky one, I have to admit. But 2 pairs would mean 4 total chromosomes, right? So that's not it. It would have to be what? B, exactly right. 2 sets. When they say 2 sets, that means hopefully they're talking about 2 homologous sets. That's not the same as saying 2 homologous pairs, because that would be a pair and a pair. That's 4 chromosomes. That's too many chromosomes. Does that make sense? So 2 sets of chromosomes, not just 2 of the same copy, 2 of the same set. In fact, I still don't like this definition. It should be 2 homologous sets of chromosomes. That's what diploid really means, if you want to get really technical. All right, so let's take a quick break. Hey everyone, welcome back from break time with Gizmo and Wicket. Where we left off, we were about to talk about chromosomal structure, and a lot of this is review from Biology 1406. Remember that the chromosomes have a centromere. We're talking about eukaryotic chromosomes, of course, have a centromere, and that is the narrow waist of the chromosome. Oh, and Wicket is here playing on the keyboard. I'm sorry if he makes some noise. Again, the centromere is the narrow waist of the chromosome. It's where attachment happens for the spindle microtubules during mitosis or during meiosis as well. Remember that eukaryotic chromosomes have ends, right? There are 2 ends to the chromosome. Let me show you what I'm talking about. Here's a chromosome. Here's that narrow waist called the centromere. And you have one end up here, and you have another end. These are your telomeres. So when I say telomere, telomeres are the ends of eukaryotic chromosomes. When I say centromere, that's the narrow waist of the chromosome. By the way, centromere does not mean it's in the center of the chromosome. There are different locations of the centromere. You could have submetacentric. You could have metacentric. This is more what you would think of as the center of the pair. And then you have telocentric. That means the centromere is kind of near where the telomere would be. And then you have archrocentric as well. That's close to the telomere. So the centromeres can be in different positions depending on the chromosomes you're talking about. Okay. And again, I have a really nice video right here at the bottom left, and I'll link it to I'll add it as a slide to this YouTube video at the top as well that you could click on. But this video, I highly recommend it. This video is my explanation of the difference between homologous chromosomes and sister chromatids. It's very, very important to understand the difference there. Hi, Wicket. Wicket's over here visiting. And trying to say hi. And again, do you remember the origin of replication? There are several origins of replication on eukaryotic chromosomes, not just one origin of replication as you see with prokaryotes. So concept check time. What would be the result if a chromosome did not have a kinetochore? So what is the kinetochore? Let's go back to here. Remember, the kinetochore is found at the centromere. The centromere is the attachment site for the kinetochore, and the kinetochore is responsible for spindle microtubule binding during mitosis and meiosis. Do you see that? So it's not that the spindle microtubules bind directly to the centromere. It's that the spindle microtubules bind to the kinetochore. The kinetochore is found at the centromere. So the answer should be, what? The answer is the kinetochore is the point at which the spindle microtubules attach to the chromosome. If the kinetochore were missing, the spindle microtubules would not attach to the chromosome, so they would not attach to the centromere. The chromosome would not be drawn into the newly formed nuclei, resulting in cells that would be missing a chromosome or have too many chromosomes, etc. So let's go into the life cycle of the cell, and the cell cycle. The cell cycle is the life cycle of the cell. Remember, in eukaryotes, you have two main parts to the life cycle. You have interphase and M-phase. Does that make sense? You have interphase. This is when the cell prepares to divide, and when the cell does its job and just does its job as a cell. And then you have M-phase. M-phase is when the cell divides. So when you're thinking of mitosis, mitosis is M-phase. When you're thinking of a cell outside of mitosis, that's interphase. So we're going to talk more about what goes on during interphase and what goes on during M-phase. And so this is the life cycle of a cell. And keep in mind from Biology 1406 as well that there are checkpoints that monitor and regulate the cell cycle. In your body, let's say, you're a eukaryote, your cells know when to divide, when not to divide, when to enter mitosis, when not to enter mitosis. So your cells know when and where is proper to divide. It's when the cells lose that control, that regulation, that the cells divide, and that's how actually you get cancer. Cancer is the result of these checkpoints, these cell cycle checkpoints, losing control and the cell dividing too frequently. Okay, does that make sense? And that's usually a cause by mutation. So let's talk about the cell cycle and these checkpoints as well. You have, this is the cell cycle. This chart represents the cell cycle. This long portion here, this larger part of the pie, this is interphase. Interphase takes up usually approximately 80% on average of the cell life cycle. So it's a long time. Interphase takes the most amount of time. There are three parts to interphase: G1, S, and G2. During G1, the cell grows. Then, if there is no signal to continue, let's say the cell is growing, right? The cell is growing during G1. If there is no signal to continue, if this G1S checkpoint is not released, then the cell exits the cell cycle and enters what's known as G0. Cells may enter G0, a non-dividing phase. Think of a cell just being a cell during this point. Like, let's say one of your skin cells is just doing its job being a skin cell, or your heart cells are just doing their heart cell job. They're not trying to divide. Nothing's trying to divide. That's what G0 means. The G0 means that the cell is not preparing to divide. The cell is just doing its job as a cell. Now, let's say that the cell does decide to divide, or it gets the signal to divide. The G1S checkpoint would then be released. The cell would proceed past the G1 into what's known as the G1S transition into S phase. S phase is also known as the synthesis phase, and this is where the DNA duplicates. So you're copying the DNA. Remember I told you each one of your cells has 46 chromosomes? Well, during S phase, you're going to copy each and every chromosome. So you're going to end up with how many chromosomes during S phase? That's right, 92 chromosomes. Does that make sense? So you went from 46 chromosomes before S phase, 92 chromosomes after S phase, and then you enter what's known as G2, another subphase of interphase. This is where the cell prepares for mitosis. Certain things happen here, like the centrioles replicate. So now you have two pairs of these centrioles, two centrioles. Also, the mitochondria divide, the cell grows some more, prepares for mitosis. Now, near the end of the G2 checkpoint, you have the end of G2, you have another checkpoint. It's called the G2M checkpoint. This is where the cell ensures that the DNA was replicated correctly and you don't have a bunch of mutations or damaged DNA. If this is the case, that everything's fine, the G2M checkpoint clears, and you enter M phase, or mitosis. And during mitosis, you have, remember, 5 subphases. You have prophase, prometaphase, metaphase, anaphase, telophase, cytokinesis. So you have all of that going on during M phase. You have mitosis plus cytokinesis during M phase. And by the way, there's another checkpoint during mitosis. It's called the spindle assembly checkpoint. This occurs during metaphase, where the cell checks and makes sure that the sister chromatid pairs are all attached to microtubules. Because let's say there's one sister chromatid pair that is not attached to a spindle. Well, that would result in problems. That would result in aneuploidy, or the wrong number of chromosomes after cell division. So the spindle assembly checkpoint would prevent that from happening. Does that make sense? So again, to review, and I have another review on this. I'll link a video with this in much more detail. But there's 2 parts to cell cycle: interphase, which consists of G1, S, and G2, followed by M phase, which consists of mitosis plus cytokinesis. By the way, mitosis is not the same thing as cytokinesis. Mitosis is its own thing. That's where you separate the DNA so that you end up with 2 genetically identical cells. Cytokinesis is its own thing. Cytokinesis is when you separate the cytoplasm into 2 distinct cells. So mitosis, separation of DNA; cytokinesis, separation of the cytoplasm, separation of the 2 physical cells. Okay, however, mitosis plus cytokinesis equals M phase. Does that make sense? M phase means mitosis plus cytokinesis. Mitosis does not include cytokinesis. Okay, hopefully that makes sense. So you got mitosis, cytokinesis, then you've got interphase G1, S, G2. Wonderful. We touched on this. Interphase has G1, S, G2. G1 is for growth. G1, S, checkpoint. This is where the cell decides whether to divide or not. This is the main checkpoint of the cell. What do I mean by that? The G1S checkpoint is the main checkpoint. This is the checkpoint. This is the point at which a cell decides, okay, am I going to divide or not? Am I going to just stay a skin cell? Am I going to just stay a heart cell? Or am I going to divide, right? Then during S phase, there's DNA synthesis. That means you copy all of the chromosomes. By the way, fun fact, before S phase, the cell does not contain any sister chromatids. Okay, did you guys follow that? Before S phase, there are no sister chromatids in the cell. After S phase, that's when you have sister chromatids in the cell. You know why? Because sister chromatids means identical copies of the same exact chromosome. Did you follow that? Sister chromatids means identical copies of the same exact chromosome. Would you have identical copies of things before you copy things? No. Okay, so a cell before S phase does not contain sister chromatids. A cell after S phase contains the sister chromatids, and it's during mitosis that those sisters separate. Does that make sense? G2, again, this is where the cell prepares for cell division. This is where you get the centrioles replicating, and the cell grows as well and prepares for cell division. And then you have the G2M checkpoint. Remember, this is the checkpoint where the cell scans the DNA, makes sure that the DNA is not damaged, that the DNA was replicated correctly, those sister chromatids were formed correctly before the cell decides, okay, let's go ahead and divide. Because you don't want to just divide if you have damaged DNA. That could result in problems like cancer. M phase, remember M phase equals what? M phase equals mitosis plus cytokinesis. But remember, mitosis does not mean cytokinesis. Mitosis is the separation of the sister chromatids, the separation of the DNA into two genetically identical cells. Cytokinesis is the separation of the cytoplasm. Mitosis means prophase, prometaphase, metaphase, anaphase, and telophase. Cytokinesis, you know, it's different between plants and animals, right? Do you guys remember this? In animals, you have cytokinesis by way of a cleavage furrow, and in plants, you have cytokinesis by way of a cell plate. So, by the way, everything I just mentioned about mitosis, watch this video here. I'll also try to throw a card up above. Watch this video here, and this video is going to be great. It's an even more in-depth look at mitosis, the steps of mitosis, and it's going to go into great detail about these steps. But I'm going to briefly touch on these steps here because, again, this should be a review for you. I shouldn't have to go through every single little step of mitosis because it is a review. It is from 1406. And if you need a more in-depth review, what do? Watch this video here. This video is me going through every little bitty step of mitosis, and that takes a lot longer. So briefly, I'm going to touch on the important things here during cell cycle. What do we have? Starts here with interphase. Interphase, what's going on? The cell grows, right? G1. The DNA gets copied, S phase. And then the centrosomes, see? The centrosomes, the centrioles, they get replicated. They get replicated. All right. Then what? Prophase. What happens during prophase? Now we've got left interphase and we've entered M phase. We've entered mitosis. Mitosis starts with prophase. During prophase, what do you need to know? You need to know that the DNA condenses. The DNA goes from spaghetti like this. You see what I'm showing you here? The DNA goes from spaghetti to condensed. Condensed. When this is called euchromatin, by the way, just in case you're wondering. This is called euchromatin or uncondensed chromatin. The DNA condenses into this heterochromatin. This is condensed chromatin. Does that make sense? I like to say it this way. DNA in a normal cell, let's say a skin cell that's in interphase, right? A cell that's just being a cell, the DNA gets unraveled, right? The DNA kind of spaghettifies is what I like to say. Does that make sense? This allows the DNA to loosen up so that you can read all of the genes and you can transcribe and translate all of the genes. Does that make sense? Or the genes that you need for that cell. But when the cell wants to divide, the cell needs to get everything in order to make sure that you've copied everything and you separate all the copies evenly. Does that make sense? Is it easy for you to get everything organized for separation if you've got spaghetti everywhere? No, because all the DNA is just going to be all tangled up in a big mess. Does that make sense? The DNA being all loose like this is great during G0 or during interphase. But when the cell wants to divide, okay, you don't want the DNA just being loose willy-nilly all over the place. There's no way you're going to divide the cell correctly and make sure this cell gets a chromosome 1 and that cell gets a chromosome 1. So do you know what happens during prophase? You need to clean up that DNA. The DNA goes from spaghetti looking to condense into these what are called as mitotic chromosomes that look like the X's you know and love, right? You see this X shape? That only occurs right before mitosis during prophase. You collect the DNA. Think about it. I have a computer here. You see all these wires? Oh, you can't see it, but there's wires everywhere, right? But if I was going to move my computer to a new house, I would probably take each wire, coil it up into a ball, and then that way I know I've got all my wires and they're not going to get all unorganized and I'm not going to leave any wires behind, etc., etc. Does that make sense? The cell is the same way. Everything's unorganized, but now it's time to separate let's coil up the DNA so that we can separate things in a clean fashion and make sure the daughter cells all get one copy of everything they should get. Okay, so what happens during prophase? And by the way, here's a video of me going into this detail again about prophase. If you want to just learn about prophase, here's a video. Again, during prophase, chromosomes condense into mitotic chromosomes. Okay, the centrosomes start moving to opposite poles of the cell. The mitotic spindle starts to form. Microtubules start to develop, start to form from the centrosomes. Prometaphase. Again, here's a video here talking about prometaphase. Prometaphase. What happens? The nuclear envelope breaks down. Look here during prophase. Do you see a nucleus? Yes. Look at this. The nucleus is still there. The chromosomes are inside the nucleus, right? Look at prometaphase. What happened to the nucleus? It's gone. The nucleus breaks down. The nucleus breaks down. This is the important thing to know about prometaphase. The nucleus is gone. The nucleus broke down into little vesicles. And look, the centrosomes are now at opposite poles of the cell. And the centrosomes, this is where the microtubules organize from. In fact, centrosomes are known as microtubule organizing centers, or MTOCs. That means that they are the regions where microtubules assemble themselves. Microtubules form from the centrosomes. Microtubules form the mitotic spindle. Those microtubules attach to the sister chromatids. And by the way, what are these X's? What are these X's? These are called sister chromatid pairs. So you see this here, what I'm highlighting right here? That's, for example, let me give you an example. This would be mom's chromosome 1. You see this? Mom's chromosome 1. So what's this on the other side? What's the other part of the X? Exactly. It's also mom's chromosome 1. These are identical mom's chromosome 1. So these are called sister chromatid pairs. This is a sister chromatid and it's paired up with another sister chromatid. So this is a sister chromatid pair. 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 So again, this would be two exact copies of mom's chromosome 1, 2 exact copies of dad's chromosome 1, 2 exact copies of mom's chromosome 2, 2 exact copies of dad's chromosome 2. And what's happening? Microtubules are attaching to the kinetochore on the centromere of the sister chromatid pairs. Did you guys follow that? Hopefully you're following all of this terminology. Again, it's review from 1406, so please do try to brush up on that. If you don't know, watch the videos from 1406. Again, the centrosomes produce mitotic spindle, which is essentially microtubules, which attach to the kinetochore proteins on the centromere, that means the narrow waist, of the sister chromatid pairs. And to what end? These sister chromatid pairs start making their way to the center of the cell. They start making their way, and that's what happens during prometaphase. The sister chromatid pairs start to move to the center of the cell. Why? Because the mitotic spindles start attaching and guiding them to the center of the cell. And then during metaphase, now the microtubules have guided the sister chromatid pairs to the center of the cell. So what do you need to know about metaphase? You need to know that the sister chromatid pairs line up at the center of the cell. So you've got an identical copy of dad's chromosome 1, and then also an identical copy of dad's chromosome 1. So you have identical copies down the center of the cell. Okay. Now, what happens during anaphase? The two sisters separate from one another. So you need to know during anaphase, the sister chromatid pairs separate from one another. Sisters separate from one another. And how did they separate? I wanted to show you something back here. Where's that picture? Yes, look here. It doesn't show you here, but I wanted to tell you that the reason these sisters are held together at the centromere, you see, this sister and this sister, so this would be like mom's chromosome 1, and this is an identical copy of mom's chromosome 1. The reason they're held together is because proteins in the center are called cohesin proteins. You may have learned this concept in Biology 1406. Cohesin proteins are holding the sisters together. So during anaphase, those cohesin proteins let go, and so the sister chromatid pairs can separate from one another. Sister chromatid pairs separate from one another. You know what that means? The same exact information that's heading towards this daughter cell is heading towards the other daughter cell. Does that make sense? Isn't this going to result in two genetically identical cells? Think about it. You had 46 chromosomes. You copied every single chromosome in S phase. Then all the copies lined up and as X's, right? All the copies lined up, and then the copies separated from one another. This means that just like if you had photocopied a 46-page lab report and then separated it equally, you're going to end up with two identical copies, right? So each daughter cell is going to end up with two identical copies of the DNA. Identical copies. So each daughter will have 46 chromosomes, and they will both have the same information. So during telophase, what do you need to know? Again, now you have identical daughter cells. So this would be mom's chromosome 1 in this daughter cell. This daughter cell also has an exact copy of mom's chromosome 1. This would be dad's chromosome 1 in this daughter cell. This daughter cell has the exact same copy of dad's chromosome 1. So you have genetically identical daughter cells. That's what you want. Genetically identical daughter cells. And what else is happening? What's this dashed line? The nucleus starts to reform. So there's a nucleus forming here and a nucleus forming here. So daughter nuclei form, and two genetically identical cells result during telophase. And usually during near the end of telophase, this is where cytokinesis begins. But again, is cytokinesis part of telophase? No. Is cytokinesis even part of mitosis? No. Cytokinesis can have a little bit of overlap with telophase, so it could be concurrent with telophase, but it is not part of telophase. And here's another outline of what I just went over. S phase, G0, G1, S phase, all the parts of mitosis. All right, so what are the genetic consequences of the cell cycle? I just told you, producing two cells that are genetically identical to each other and with the cell that gave rise to them, the mother cell. The newly formed cells contain a full complement of chromosomes. What does that mean? They each possess, if you're a human, each of the daughters would have 46 chromosomes, just like the mother cell had 46 chromosomes. Each newly formed cell contains approximately half the cytoplasm and organelle content of the original parent cell. That makes sense. You just split the cytoplasm in half. That's why the cell needs to undergo G1. It needs to grow again, right? The cell needs to grow again before it can divide again, because it's left with approximately half the cytoplasm of the original mother cell. So concept check time. Which is the correct order of stages in the cell cycle? Is it G1S, prophase, metaphase, anaphase? Is it GS, G1? No, that's already wrong, isn't it? G1 is before S, so that's wrong. Is it prophase S? No, that's already wrong. S and G1 is first, then S, so this is wrong. S, G1, no, that's wrong. It has to be A, right? So G1 happens before S, and then actually G2 happens, and then prophase, so it's A. So what do we see here? We're seeing another overview, an overview of mitosis. Remember, here you have interphase, and then you have M phase here, M phase. This is showing you, though, the number of chromosomes in the cell. The cell during G1 would have 4 chromosomes. If this was a human, it wouldn't have 4 chromosomes. How many chromosomes would a human cell have during G1? 46, right? 46 chromosomes. Then during S, what happens? Synthesis stage, right? Copy all the chromosomes. How many chromosomes are you going to have during this? You're going to have here it's 8, but 46 becomes 92. Okay, during G2 you have 92. Then what? During prophase and prometaphase, 92. During metaphase, 92. Anaphase, 92. And then telophase cytokinesis, how many chromosomes are in each cell? It would be 46. You see, so you're kind of seeing how the process works here. And by the way, look, what are these X's? Remember this? Look here. What are these X's? These are the sisters, aren't they? The sister chromatids. Are there X's here in the original cell? No, because remember what I said? The sister chromatids only form after S phase. There are no sisters before S phase. There are only homologous. Here, there are homologous chromosomes in G1. You know why? Because you have a mom's chromosome 1 and a dad's chromosome 1. Those are called homologous chromosomes. But you don't have sister chromatids in G1. Sister chromatids means the exact same thing. So you have 2 copies of mom's chromosome 1, 2 copies of dad's chromosome 1, right? So now let's go on into meiosis. But before we do, another quick break time with Gizmo and Wicket, so I can, catch my breath a little bit. And we'll come back and we'll talk a little bit about meiosis. All right, welcome back from break time with Gizmo and Wicket. Let's carry on with our concept of meiosis. Again, I have a really nice video on meiosis, which I'll show you when we get there. We have a nice link, and I break it down step by step with drawing. So if you need a refresher, you can always go to that video. But let's try to see what we can cover here. Again, you have interphase. With meiosis, you do have interphase. It does consist of G1, S, and G2, just like before. Remember, DNA synthesis occurs during S phase. However, there's only one interphase. There is no interphase between meiosis 1 and meiosis 2. You have interphase, then you jump into meiosis 1. Meiosis 1 is called the first separation. Okay, it's the first time the cell divides. Here is where the homologs separate. This is important to know. During meiosis 1, the homologs separate, and you reduce the number of chromosomes by half. Okay, the homologs separate. This is also called reduction division. The reduction of the chromosome number from diploid to haploid. Meiosis 1 is called reduction division because you go from diploid cells, a diploid cell, I should say, to 2 haploid cells. During meiosis 2, this is the separation of the sister chromatids. And during this process, this is called equational division, where you're simply having the number of chromosomes. During meiosis 1, you have the number of chromosomes, but this takes you from diploid to haploid. That's why it's called reduction division. During meiosis 2, you're still having the number of chromosomes, but you're not changing the ploidy of the cell. You're not going from diploid to haploid. You're going from haploid to haploid. Okay, again, I'll reiterate this when we get to it, and I have videos that, make this concept more clear if it's fuzzy. So again, during meiosis 1, you have your original diploid cell. This would be called a germ cell. G-E-R-M, germ cell. A germ cell is a specialized reproductive cell inside of your reproductive organs, the testes or the ovaries, right? The germ cell is the cell that will give rise to the sperm or the egg. Okay, so this germ cell is a diploid cell. It's 2N. Remember 2N from Biology 1406? 2N means diploid, 2 homologous sets of chromosomes. This diploid cell undergoes meiosis 1, So now you have 2 cells, right? Meiosis 1, you end up with 2 cells. And what's separated? Look, the homologs separate. So look, these 2 chromosome 1s from mom go to this cell. These 2 chromosome 1s from dad go to this cell. You see that? You see what happened? The homologs separated. Mom's information separated from dad's information. That's what we mean by the homologs separated. So here again, both of dad's chromosome 2 went to this cell. Both of mom's chromosome 2 went to this cell. So this is why it's called reduction division, because even though these 2 cells have technically 2 sets of chromosomes, because they have 2 chromosome 1s and 2 chromosome 2s, remember, because they don't have 2 homologous sets of chromosomes, they are not considered diploid. So even after meiosis 1, this is important for your exam, even after meiosis 1, even though the cells technically have 2 sets of chromosomes, because they don't have homologous sets of chromosomes, or 2 homologous sets of chromosomes, they are not considered diploid. That's why there's an N, right? Just an N, not 2N, but N. Remember what I said earlier? You can't just have 2 copies of everything and call yourself diploid. It doesn't work that way. You need 2 homologous sets of chromosomes to be considered diploid, right? So if this cell had a red and a blue chromosome 1, that would be homologous, that would be diploid. But because it only has 2 red ones, that doesn't qualify as diploid. That qualifies as having 2 of the same thing. Does that make sense? Awesome. So that is why meiosis 1 is called reduction division, because you're going from 2N to 1N, right? You're reducing the ploidy of the cell from diploid to haploid. And then what happens during meiosis 2? These sister chromatid pairs, you see these sister chromatid pairs line up, and then they separate. And so you separate what during meiosis 2? The sisters separate during meiosis 2. The sister chromatids separate during meiosis 2. And this is called equational division, because you went from 4 chromosomes here to 2 in each cell. Does that make sense? So what's separated during meiosis 1? The homologs. That's the reason for the ploidy change. That's the reason for reduction division. What's separated during meiosis 2? The sisters did, and that's the reason why it's called equational division, because it was a simple matter of going from 4 to 2. Okay, hopefully this makes sense. If not, I've got a nice video I'm going to show you from 1406 that should make it all clear. And again, let's break it down about what's happening during prophase 1, what's happening during all of meiosis. Because remember here, interphase has G1, and G2. Then meiosis 1 has its own prophase called prophase 1. It has metaphase 1, anaphase 1, and telophase 1. Then you have meiosis 2, which has its own prophase. It's called prophase 2. And then it has its own metaphase, metaphase 2, and anaphase 2, and telophase 2. So we need to break down what's happening during each and every step. What's happening during prophase 1 of meiosis 1? This is important. You need to know. During prophase 1, synapsis occurs. What does this mean? The homologous chromosomes pair. In fact, you could say this. One pair of sister chromatids will pair with the homologous pair of sister chromatids. Does that make sense? So another way of thinking about it very simply is that both of dad's chromosome 1 is going to attach to both of mom's chromosome 1. Think about it that way. That's what we mean. So it's going to be like 4 chromosomes. So imagine if these 2 represent both copies of dad's chromosome 1, and these 2 represent both copies of mom's chromosome 1. This is sister chromatid pair of dad's chromosome 1, sister chromatid pair of mom's chromosome 1. If they came together, that's synapsis. Now you have 4 total chromosomes, and that is called a tetrad. Tetra means 4. Tetra means 4. Tetrad means the 4 chromosomes all together. This is 4 sister chromatids, 2 homologous chromosomes. Then what happens? Do you remember in Biology 1406 you have crossing over? Because the tetrads formed, 2 of those homologous chromosomes were able to cross information, physically exchange information between one another, increasing genetic variability. So let me show you what that entails. Let me show you how that works. So let's start with this. Meiosis 1. Meiosis 1. What happens? Chromosomes begin to condense. The spindle forms. Remember, the centrosomes move to opposite poles of the cell. That's where the microtubules start to form. Microtubules form. Okay, they move to the opposite poles of the cell. The nucleus is still there. The DNA starts to condense. Remember, this is a recurring theme. The DNA is like spaghettified, right? Like spaghetti. But it's time to separate stuff, isn't it? So you need to condense. You need to tie up your cords. You need to get stuff ready for division. The euchromatin gets condensed into heterochromatin. These structures called mitotic chromosomes. And then what happens? The sisters pair up, right? So late in prophase, pairs of homologs form. Homologous chromosomes pair. But before the homologous chromosomes pair, the sisters pair, right? Sisters. So mom's chromosome 1 is going to find its exact copy of mom's chromosome 1, and both mom's chromosome 1s are going to attach. You see this red X? You see this red X? Those are the sisters. And do you see the blue X? Those are blue sisters. So the red X, that means like mom's chromosome 1, right? 2 copies of mom's chromosome 1. That's the red X. Those are the sister chromatid pair of mom's chromosome 1. They find the homologous pair. That means the 2 blue ones. They find the 2 blue ones. The 2 blue ones would be 2 copies of dad's chromosome 1. That means 2 sister chromatids of dad's chromosome 1. So what happened? Both of mom's chromosome 1 found both of dad's chromosome 1 and formed a 4 tetrad, which means homologous chromosomes paired. That's what they mean by saying homologous chromosomes paired. Technically, homologous sister chromatid pairs paired, if you want to think about it that way. So you got homologous chromosomes pair as tetrads through a process known as what? Synapsis, right? And then what happens? Crossing over. One of these red ones and one of these blue ones, one of these red sister chromatids and one of these blue sister chromatids will physically exchange tails, right? Physically exchange ends. And this is very, very important for you to understand, because in a subsequent chapter, we're going to be talking about crossing over a lot. And this is what crossing over means. You exchange genetic information between one of these 2 sisters and one of these 2 sisters. So what you end up with is one of the red chromosomes, one of mom's chromosome 1s, ends up with a little bit of dad's chromosome 1, and one of dad's chromosome 1 ends up with the little bit of mom's chromosome 1. You physically exchanged DNA. Does that make sense? You physically exchanged DNA. That's what crossing over means. And again, I think I put this video here. This video, by the way, this video in your notes will take you to a much more in-depth explanation about meiosis, where I draw every step on the board and explain every step. Step-by-step review of meiosis again, if you need that refresher. Okay. Again, hopefully, this is review. If not, please watch those videos or read that chapter in the book more closely. So now what have you done? So again, you need to know, during prophase 1, what happened? The DNA condensed, tetrads formed, which means homologous chromosomes paired through synapsis. Tetrads formed. And what else happened? Crossing over happened. Why does crossing over happen? to increase genetic variability. Because look at this. After crossing over, are any of these 4 chromosomes the same anymore? You see the 2 blue ones and the 2 red ones, are any of them identical anymore after crossing over? No. After crossing over, every one of these is now different. Every one of these sister chromatids is different. There's no, you've shuffled the genes around. You've increased genetic variability. That's what that means. Okay. Then in metaphase 1, what's lining up at the center of the cell? You need to know this for the exam. What's lining up at the center of the cell? The tetrads line up at the center of the cell. Another way of saying that is the homologs line up at the center of the cell. Another way of saying that is the homologous pairs of chromosomes line up at the center of the cell. I know there's a lot of redundancy, and there's a lot of ways of saying this correct. And then during, again, this is metaphase again. During anaphase, what separates from what? You need to know this. What's separating from what? The homologs separate. The homologs separate. What does that mean, homologs? Remember, every time I say homolog, you should think moms versus dads, right? Mom's information is heading one way, dad's information is heading the other. That's what homologs separate means. Does that make sense? Both of mom's chromosome 1 is heading to this cell. Both of dad's chromosome 1 is heading to the other cell. That's what I mean by saying homologs separate. The sisters didn't separate. The homologs separate. And by the way, what does this result in? This results in 2 genetically different cells, right? If both of the red ones are heading this way and both of the blue ones are heading this way, are these going to be genetically identical cells? No. These are genetically different cells. Does that make sense? 2 different genetically different cells. And not only that, but remember, meiosis 1 is considered what? Reduction division, because even at the end here, at telophase 1, these are already haploid cells. This is a haploid cell, and this is a haploid cell. Even though they technically, you could argue they have 2 sets of chromosomes, but it doesn't matter if you have 2 blue chromosomes. You can't have 2 similar sets of chromosomes and call yourself diploid. You have to have 2 homologous sets of chromosomes to be diploid. So again, what happens during anaphase? During anaphase, the homologs separate. What happens during telophase? Daughter nuclei form, and you end up with 2 genetically non-identical haploid cells. Then we're ready for meiosis 2, right? What happens during prophase 2 of meiosis 2? What happens? The DNA condenses, right? The DNA condenses, and the DNA forms sister chromatid pairs. Sister chromatid pairs. By the way, if you want to be very specific, if you want to be highly accurate, what you would call these are not sister chromatids. You would call them non-identical sister chromatids. Does that make sense? Non-identical sister chromatids. Why would I call this X a non-identical? Look at this X right here that I'm highlighting. Why would I call this a non-identical sister? Well, because it's still a sister because it shares most of its DNA. But remember the blue tail due to crossing over? Well, that makes it a little bit different. That makes these 2 not identical, right? So that's why they're called non-identical sister chromatids. You know, if you want to be very specific, that's what you would call them. I still sometimes refer to these as sister chromatids, but just be aware that if these are sister chromatids during metaphase 2, they're non-identical sister chromatid pairs. So what lines up at the center of the cell during metaphase 2? The correct answer would be non-identical sister chromatids line up at the center of the cell. And then what happens during anaphase? What separates from what? Non-identical sister chromatids separate from one another. And then what happens during telophase? Now you end up with daughter nuclei, right? The nuclei forms again. The DNA can decondense. The DNA can decondense. Daughter nuclei form. Now, are any of these 4 cells identical genetically? Look here. Are any of these 4 cells going to be genetically identical? No. That's what you need to understand. The result of meiosis is 4 genetically non-identical cells called the gametes. So this is going to become a sperm cell. This is going to become a sperm cell. This is going to become a sperm cell, and this one will become a sperm cell. None of the 4 sperm cells are going to have the same exact genetic material. All 4 of them are genetically distinct. Okay? All 4 of them are genetically distinct. All right, again, during meiosis 1, metaphase 1, homologous pairs of chromosomes align, tetrads align down the center of the cell, right? You could write tetrads align down the center of the cell. The homologs align down the center of the cell. Anaphase 1, the homologs separate from one another. Telophase 1, you have 2 genetically non-identical haploid cells. And then you have, before meiosis 2, you have a short thing called interkinesis. We're not really going to go into that. And then you have prophase 1. What happens during prophase 2? What happens during prophase 2? The non-identical sister chromatids condense and pair up. What happens during metaphase 2? The non-identical sister chromatids line up at the center of the cell. What happens during anaphase 2? Non-identical sister chromatids separate from one another. And telophase 2, the daughter nuclei form, the DNA starts to decondense, and you end up with 4 genetically non-identical cells. All right, and what are the consequences of meiosis and genetic variation? 4 cells, not just 2. Remember, during mitosis, you ended up with 2 genetically identical cells. During meiosis, you end up with 4 genetically non-identical cells, genetically distinct cells. Chromosome number in each cell is reduced by half. The new cells are haploid. So think about it this way. You started with 46 chromosomes in the germ cell. You started with 46 chromosomes in the germ cell. So let me show you here real quick. You started with 46 chromosomes in the germ cell. That's separated into, if this is meiosis, actually, the germ cell would be back here, 46 chromosomes. And then remember, you copied all the DNA during S phase of interphase. So you enter meiosis with how many chromosomes? Not 46, but 92. So you have 92 chromosomes in meiosis. The cell divides 46, 46. And then the cell divides again without copying anything. So you end up with 23 chromosomes, 23 chromosomes, 23 chromosomes, 23 chromosomes. Does that make sense? The original germ cell had 46, but you copied all those 46 during S phase. Now you had 92. Then you entered meiosis with 92. Cell divided to give you 46, 46, even though they're haploid. And then divided again to give you 23 in each of these daughter cells. So each sperm cell would have 23 chromosomes. Each egg cell would have 23 chromosomes. All right. Again, this is showing you how crossing over produces genetic variation, because when you have crossing over, it's impossible to have genetically identical cells at the end of crossing over. By the way, you need to know for the exam that there are 2 ways of increasing genetic variability or genetic variation during meiosis. One way is crossing over, which occurs during what? Prophase 1. During prophase 1, crossing over increases genetic variability. Does anyone remember what the other step of mitosis is that increases genetic variability? That's this. This is called independent assortment. In fact, I don't know why I don't have a slide title for this. I should, but you should label this slide independent assortment. Independent assortment. This is the other major way that there's genetic variability introduced during meiosis. And when does this happen? So look, this is the cell, which has 3 chromosomes. Let's say mom's chromosome 1, mom's chromosome 2, mom's chromosome 3, right? Dad's chromosome 1, dad's chromosome 2, dad's chromosome 3, right? You copy all the DNA. Here's all the DNA copied. And then you do metaphase 1. This here represents metaphase 1. Metaphase 1. What's lining up at the center of the cell during metaphase 1? Do you guys remember? The tetrads, or you could say the homologs, line up down the center of the cell with metaphase 1. Now what you need to understand is these tetrads can organize themselves independently. It's random. It's random. However way this tetrad aligns itself has no bearing on how this tetrad lines up, has no bearing on how this tetrad lines up. So essentially what I'm trying to say is, if you have, let's say, the 6 different sister chromatid pairs like this, you see? 6 different sister chromatid pairs, then they could line up this way, as shown in this first drawing. They could line up this way. They could line up this way. They could line up this way. You see that? They could line up any one of these ways. And because of that, you're going to get different results. You see? You're going to get different results in the gametes. You see this? You're going to get different gamete outcomes. So if you have 3 different chromosomes, those homologs could separate how many different ways? How many different ways? 8, right? It's 2 to the power of 3. 2 to the power of 3 is the number of different ways that those homologs could separate during, or I should say right after metaphase 1. And that's going to give you more genetic variability. But do humans have only 3 different chromosomes, like chromosome 1, 2, and 3? Do humans only have 3 different chromosomes? Think about that for a minute. How many different chromosomes do humans have? Different chromosome numbers. Humans have 23 chromosomes, don't we? Don't we? We have 23 chromosomes. So we don't have 3 chromosomes. We have 23 chromosomes. If we only had 3 chromosomes, independent assortment could shuffle our chromosomes 8 different ways. But we actually have 23 different chromosomes. So let's grab the calculator app. All right. And I need I need my handy dandy scientific calculator here. Again, 2 to the power 3 was 8, right? This is the number of ways that your chromosomes could shuffle during independent assortment if you only had 3 different chromosomes. But we have 23 chromosomes, don't we? So let's figure that. 2 to the power 23. Now we were talking 8.4 million. 8.4 million, you guys. Think about this for a second. You have 8.4 million different ways that your chromosomes could line up during interphase. During metaphase 1 of meiosis. That's a lot of genetic variability. Think about that. That means each one of the sperm cells your body produces, or each egg your body produces, there was a 1 in 8.4 million combination there. So the odds of your body shuffling those chromosomes in that particular arrangement again is 1 in 8.4 million. Does that make sense? It's baffling to think about, but only 1 in 8.4 million sperm cells can shuffle their chromosomes around the same way. And does that account, does this 8.4 million account for crossing over? Think about that for a second. Or is this just referring to, are we just referring to independent assortment when we're talking about 8.4 million? Are we talking about, we're just talking about independent assortment, aren't we? We're not talking about crossing over. Crossing over doesn't happen the same way twice either, right? Does that make sense? So, you know, the odds of you making the same sperm cell twice is 1 in 8.4 million with regard to independent assortment. But once you factor in crossing over, then it's astronomically high. The odds of you making the same sperm cell genetically the same sperm cell, or genetically the same egg cell twice, is astronomically low odds, right? I don't even know if that's truly been factored ever, if that's ever been answered, what the odds are of making two sperm cells genetically identical, 100% identical. It would be in the billions of odds, or trillions maybe, right? And that's not just for the sperm cell, but also for the egg cell. So think about it. This is fantastic, fascinating stuff, because the whole point of meiosis, is to increase genetic variability. Because you don't want to make all of your offspring clones of yourself. You want to introduce genetic variation. Why? Because that's going to increase the odds that your children will be able to adapt and evolve and to persist in the face of, let's say, a new virus or a new threat to humanity. You have more robust populations when there's more genetic variability. When you have less genetic variability and everyone's a clone of one another, well, that's the best way to go extinct. The best way to go extinct is to just have clones of the same person. You know why? Because if something is going to kill me and everyone is a clone of me, well, then we're all going to die. But if there's a lot of robust genetic variability in the population and people aren't like me, people are, have their own genetic backgrounds, well, then maybe there will be a virus in the future. And that virus maybe it kills me, but it won't kill you. Does that make sense? More genetic variability, more chance of survivability. And that's why meiosis has evolved over time to increase the genetic variability as much as humanly possible. Just think, if the odds of you, think about the odds of you being born you, right? Your dad's sperm had to have this 1 in 8 million combination. Your mom's egg had a 1 in 8 million combination. So a 1 in 8 million sperm, a 1 in 8 million egg, and again, that's not even factoring in the crossing over that happened in the sperm and egg to make you, right? So think about how truly unique you are genetically, right? How likely would it be for your parents to have another child at a later date and have your exact DNA? Is it possible for your parents to have another child at a different date with your exact DNA? It is technically possible. I mean, you are the result, right? So why couldn't it happen again, right? But do you know the odds of that happening are in the fractions of a trillionth, right? Does that make sense? So you'd be more likely to win the mega lottery 10 times in a row and then get struck by lightning than, the odds of your parents having you again. It's so rare that it's probably never happened in the history of all humanity. Isn't that interesting to really think about how truly genetically unique you are? It's fantastic. So, and that is a side effect. That is as a result of this gene shuffling that happens, this genetic variability that's introduced during what? Crossing over during what? Prophase 1, and independent assortment during what? During Metaphase 1. Does that make sense? Awesome. I know that's a long story, but very fun to think about. So again, concept check 5, concept check time. Which of these following events takes place in Metaphase 1? We just talked about this. Crossing over? No. The chromosomes condense? No. Okay, wait. Crossing over happened when? Crossing over happened during Prophase 1. Chromosomes condense? No, that happened during prophase 1. Homologous pairs of chromosomes line up on the metaphase plate. Yeah, that's true. Individual chromosomes line up. No, let's say C. Yeah, we got it. Got them. All right. All right. So again, here's an overview of mitosis versus meiosis. Okay. Oh, this was that term I was trying to tell you about earlier. Cohesin proteins. Remember cohesin proteins? These are the proteins that hold the sisters together, that hold the sister chromatids together. These are the proteins that release when anaphase occurs. Okay. So you see, during mitosis, the sisters are held together by cohesin proteins, and then during anaphase, those cohesin proteins let go, and that's why the sisters let go of one another. During meiosis, same thing. You have cohesin along the chromosomes, holding them together, and then during anaphase, the cohesin proteins break apart. All right, and then just to explain how the sperm and the egg form, this is called spermatogenesis, that's male gametogenesis, and oogenesis, which is female gametogenesis. So spermatogenesis is how the sperm forms. Let's go through this together real quick. Spermatogonia, remember, these are your germ cells. Remember, the germ cells are the reproductive specialized cells in your reproductive organs that give rise to the sperm and the egg through the process of meiosis. Spermatogonia in the testes can undergo repeated rounds of mitosis, producing more spermatogonia. So spermatogonium are kind of like stem cells in that way. A spermatogonium may enter prophase 1, becoming a primary spermatocyte. This is your primary spermatocyte. It's diploid, 2N. Each primary spermatocyte completes meiosis 1, producing 2 secondary spermatocytes. So this would be before meiosis 1, and then you have after meiosis 1. After meiosis 1. Are these cells diploid? No, they're haploid, 1N. So just realize, after meiosis 1, the cells are technically haploid. Then we're ready for meiosis 2, aren't we? That then undergo meiosis 2 to produce 2 haploid spermatids each. So each of these makes 2 haploid spermatids. And then these spermatids mature into 4 sperm cells. Are any of these sperm cells identical genetically? No. Are these sperm cells diploid or haploid? Haploid. How many chromosomes do these sperm cells possess? 23. So each sperm cell contains one set of 23 chromosomes. Does that make sense? Now let's compare and contrast that to oogenesis. This is the production of the egg, the female egg. Oogonia in the ovaries may either undergo repeated rounds of mitosis, producing additional oogonia, or they could enter prophase 1, becoming primary oocytes. Here's a primary oocyte, which is diploid. Then each primary oocyte completes meiosis 1, producing a large secondary oocyte and a smaller polar body, which disintegrates. Okay, so look here. This oocyte, this secondary oocyte will persist. It's haploid already, obviously, because meiosis 1 results in 2 haploid cells. This cell will go on to become the egg. This cell is called the polar body, and it actually disintegrates. It's lost. It doesn't become an egg. Now you're ready for this cell to undergo the meiosis 2. Meiosis 2. And the secondary oocytes completes meiosis 2, producing an ovum and a second polar body, which also disintegrates. This poor secondary polar body will disintegrate. So you only end up with one ovum, one egg. You only end up with one egg from one oogonium, whereas from sperm you end up with 4. Now you technically should have ended up with 4 cells, but remember the poor polar bodies kept disintegrating, right? That's what happens. So then the sperm meets the egg, and that is fertilization to give you the diploid zygote. So we just touched on this, meiosis in animals. Spermatogenesis, that's the sperm production. Oogenesis, this is female gamete production. And that leads us to the end of our story time. I know this was a long chapter with a long story time, but hopefully you learned a lot. Hopefully, this is all review, or most of it was review. If not, like I said, I have all those nice links to more detailed chapters on mitosis, meiosis, the difference between sister chromatids and homologous chromosomes. We can do it. Let's get through it. If you have any questions, ask them in the comment box below, and I'll catch you guys next time.
Dr. D, Dr. D, Dr. D. 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 Dr. D, Dr. D, Dr. D.